Okay, so let's take a look now at lipid storage in adipose tissue. So the primary function of adipose tissue is to be a reservoir for long-term storage of energy. Now we've seen that in the bloodstream we have a nice system to be able to remove fats in the form of chylomicrons or VLDL, lipoprotein particles that are our triglyceride carriers. And by having a tethered system of these factors binding to the HSPG molecule, we localize this substrate to lipoprotein lipase, which is also tethered to the HSPG, and of course our ApoE accessory protein is mediating this interaction, and then ApoC2 is our activator of lipoprotein lipase. The net result of this activity in the circulation is to deliver fatty acids into the extrahepatic tissue. Now in the case of adipose, the function of adipose, of course, is to take this fatty acid and to store it for long-term need. So let's look at what's going on inside a fat cell during this event. Now remember, this is a fed state we're primarily talking about here. So fed state means elevated insulin levels. Insulin will bind to its receptor and trigger the movement of GLUT4 containing vesicles to the cell surface. So this is analogous to skeletal muscle's response to insulin. So insulin will cause the mobilization of these vesicles and place GLUT4 in the plasma membrane. Now recent evidence has also indicated that a fatty acid transport protein, FATP, which this particular one is called CD36, is also found in the same vesicles that harbor the GLUT4 transporter. So in essence, when insulin signals causing the movement of these vesicles to the plasma membrane, we not only insert GLUT4 to the plasma membrane, but we also insert a fatty acid transport protein. Now under these fed conditions, the fatty acids that's released by the lipase activity will pass through the transporter, and the first activity, of course, is the attachment of a coenzyme A to the fatty acid by the fatty acyl-CoA synthetase. This enzyme serves, of course, to trap the fatty acid so the reverse transport will not work. It also charges the fatty acid so that subsequent reactions will have the energy to proceed. Now we have our fatty acids charged with CoA in the cytosol. Now the question is, how are we going to store this? Well, in order to do that, we need glucose from our diet, which is going to pass through the GLUT4 transporter, which has been exposed by insulin signaling. And glucose here will undergo oxidation through glycolysis, and we are going to bring this down to the level of dihydroxy acetone phosphate plus glyceraldehyde 3-phosphate. Now the glyceraldehyde 3-phosphate, of course, can continue on in glycolysis. The next step of this reaction, of course, with the dehydrogenase enzyme to produce 1,3-bisphosphoglycerate is going to generate NADH. This will be used with DHAP. Recall DHAP can be reduced to form glycerol 3-phosphate. Once we have glycerol 3-phosphate, now we have the precursors for triglyceride synthesis. So recall the glycerol 3-phosphate molecule. The next step of this reaction is to take a fatty acyl-CoA, attach it to the one carbon. We will take another fatty acyl-CoA, attach it to the new carbon, the second carbon, and then what we end up with here is phosphatidic acid. Now we examined this pathway before, that once we form phosphatidic acid, which is essentially glycerol 3-phosphate with two fatty acids on the carbons 1 and 2, this can branch either to membrane synthesis to form a phospholipid, or this can form a triglyceride. The fact that adipocytes do not undergo replication, the regeneration of material for the plasma membrane is of low need, and so this pathway is reduced, and the triglyceride synthesis pathway, which is for storage, is the predominant pathway. So to do this, we remove the phosphate, we add a third fatty acid onto the number 3 carbon, and we have our triglyceride structure. Now the triglycerides inside the adipocyte are stored in what's called a lipid droplet. As you can imagine, this is a highly hydrophobic structure and must be protected from coming in contact with the plasma membrane, or we could have solubilization of the membrane and cell lysis. And so there is another protein that essentially studs around the lipid droplet and shields the lipid droplet from the plasma membrane and other components in the cell that could be damaged because of the hydrophobicity. This particular protein is called perilipin. Peri around, lipin, lipid, around the lipid. So this perilipin essentially is a coat around the lipid droplet to protect the adipocyte. Now all of this is regulated, and the primary regulation we need to take a look at is called PPAR gamma. And this stands for the peroxisome proliferator activated receptor. The ligand for this receptor, this is an intracellular receptor, and so the ligand for this receptor is thought to be a fatty acid. So upon entrance of fatty acids, the fatty acid binds to PPAR gamma, and this translocates to the nucleus. Once it reaches the nucleus, it dimerizes with another transcription factor called the retinoic acid X receptor, RXR. Once we form this dimer, now we have a transcription factor that can bind to a promoter element that is called PRE for peroxisome proliferator activator receptor, or PPAR gamma response element. PPAR gamma response element. Now, being a master gene regulator, master transcription factor, this is going to drive the expression of multiple genes. These genes include the genes for LPL, lipoprotein lipase, which is involved in increasing fatty acid liberation from our lipoproteins for fat storage. It also increases the expression of CD36 for increased fatty acid entrance into the cell. It increases the expression of GLUT4 for increased glucose oxidation, but also increased DHAP product formation so that we can form the glycerol 3-phosphate backbone to form our triglycerides. It also increases the expression of small protein called AP2, which is a fatty acid binding protein which escorts fatty acids through the cell once they're trapped by the synthetase enzyme to the site of synthesis for triglycerides. The other important protein PPAR gamma upregulates is perilipin, and this is of course for protection once the triglyceride lipid droplet is formed. Now, the actual ligand for PPAR gamma that activates this entire sequence is not completely understood, but what's important to understand here is that our glitazones, class of drugs that are anti-diabetic, act at this point. The glitazones will bind PPAR gamma and mimic the activation of a fatty acid. So thus, by having the glitazone activate PPAR gamma, what we are essentially doing is increasing the uptake of fatty acids by adipocytes and storage into triglyceride structures. This will reduce the plasma levels of lipids and prevent lipid-mediated insulin desensitization. So the glitazones act by increasing lipid storage and decrease by this insulin resistance.